Hello everyone, and welcome to this session on SLE, systemic lupus erythematosus. Okay, what is the full form? It's a bit tough, but you have to remember systemic, as it is a systemic disorder. It is not a local disorder like one hand or one feet is not involved. It is a systemic disorder, occurs throughout the body. Then you have lupus, okay, and erythematosus. Erythematosus. So it is basically a disorder, a autoimmune disorder. So if I talk about autoimmune disorder, we have two cartoons here, just to give you a boost. So these cells are telling that intruder destroy, but this cell is just a diseased cell, but it is trying to pretend that it is a cell of our human body only. So it's telling, but I am one of you. Okay, but they are not listening to their lies. But our body is trying to uh, prevent a autoimmune disorder from happening, but the cell is exactly same as our body cell. Okay, so autoimmune disorder is a type of disorder in which the own cells of our body is basically attacking you, right? So this is the autoimmune disorder in a nutshell, and this is the cartoon which you can understand. Okay, a reel you must have seen in Instagram also. Baby was pulling his hair and crying. Okay, that thing is there. That babies tend to pull their own hairs and then cry due to that. So our body is harming ourselves, right? When our body tries to harm ourselves, it is called as the autoimmune disease. Autoimmune. So you know about immunity. Immunity is basically helping the body to destroy pathogens, but autoimmunity means our own body is basically acting against us, right? So this is the autoimmune disease. So systemic lupus erythematosus. I'm sure you must have remembered the full form till now, but you have to do. So SLE is basically an autoimmune disease. It's the first point, right? Okay. So you can see over here. Okay, we'll move to this. So, whenever I talk about an autoimmune disease, we can have many causes. A autoimmune disease like एक disease का एक cause नहीं होता है. If you have a disease, you generally think that only one cause will cause the disease, right? But here, if I talk about autoimmune disease, then we can have many causes, many factors causing it. Many factors will be causing it. Okay. So, what are the factors here? We have to discuss. They ask you, what are the factors causing this type of autoimmune disorders, right? So you can have three type of factors. Whenever I tell that it is an autoimmune disease, you just remember. Okay, whenever I tell you that it is an autoimmune disease, you have to think of genetics, G, genetic factors, environmental factors, E, and immunological factors, I. Genetics, environment, and immunological factors, right? Three type of factors are important whenever you tell about a autoimmune disease. These three factors total, if they contribute to you, then autoimmune disease is caused, right? So let's talk about some genetic factors. Okay, I don't know if you know or not, but we have something in our body which is known as HLA. 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 Okay, so HLA, we have a gene, and a particular position in gene is called as a locus. Particular position, right? So here in a person who has HLA DQ type, a person who has a HLA DQ locus, HLA DQ locus, right? And the allele, allele, you know, in a gene it is there, allele, a specific allele 1501 slash 0301, 1501 slash 0301. If these things are present in a person, if these things are present in a person, then there is increased production of autoantibodies, right? If a person has HLA-DQ locus in allele 1501 slash 0301, very easy to remember, 1501 slash 0301, a person is more susceptible to autoimmune disease like SLE. Understood? This much is clear. This is the first point. In the genetic factors, I am discussing about genetic factors. Okay, you can just give a heading genetic factors. And in the genetic factors, I told you the first point is this HLA-DQ locus and allele 1501 slash 030, right? Okay. The second thing in genetic factors only is what is this? You are beautiful, thank you. What are this? Compliments, right? But this complement is a bit different. Complement. You all know about complement system. Complement system is basically some liver produces some molecules which are called as complement molecules. These complement molecules are basically helping in preventing different type of disorders by killing the pathogens. Simple. So when we have complement deficiency, we have some complements like C1, C2, C3, okay, C1, 2, etc. Right. So whenever we have complement deficiency, whenever you can have complement deficiency, and that of specific complements C2, C4, C1Q, C2, C4, C1Q, 2, 4, and 1, 1, 2, 4. If these factors are deficient in the body, these factors are not there in the body, you can say that complement factor deficiency also leads to SLE, right? So we discuss genetic factors. Genetic factors, what are the genetic factors? Just tell me. Once the HLA-DQ predisposition, allele 1501, 0301, complement deficiency C2, C4, and C1Q. Done? Okay. Genetic factors are done. Then we will talk about some. I told you G is done. This G is done. Environmental factors. What are the okay, let's come to E. These are the environmental factors. I told you genetic factors. Second heading is environmental factors. So you can see 1 and 2, these two diagrams. Right. So the person is asking why are wearing sunglasses, and this person is telling that to protect against ultraviolet light. Ultraviolet. He's not telling violet, he's telling violent, because UV light is very dangerous, right? So he's telling that don't you mean ultraviolet? So this person is being punched by the UV light, because UV light is finding the person cool who told ultraviolet, right? Because UV light is a violent power, right? Whenever you have UV light, this UV light, where you get UV light? From sun, right? From sun you get UV light. So whenever you get UV light, it will increase the chances of causing SLE, right? So this was a cartoon of UV light. So UV light exposure is a very important factor causing SLE, right? Second thing, what is the person drinking? You can say alcohol, but this was a cartoon of drug. So I was not discussing about this type of drugs. Right? What persons abnormally take the different type of morphine, opioid, not this type of drugs. But we have three drugs. Just remember, okay, three drugs: HS, hydralazine, sulfur drugs. Okay, hydralazine, sulfur drugs, and procainamide. These three drugs, so just remember that these are causing your SLE, right? And this type of SLE, which is due to the drugs, is called as DLE. SLE was systemic lupus erythematosus. DLE, what you can remember? DLE, what must be DLE? Drug, drug induced lupus erythematosus. Then DLE. So these drugs you can also you can write isoniazid, isoniazid. Okay. You must have heard about SIP drugs, right? In pharma, SIP drugs, sulfonamides and sulfa drugs, hydralazine, isoniazid, procainamide. The SIP drugs are basically causing this disorder SLE, right? So you got this type of concept. These things are done. So I talked about genetic factors. I talked about environmental factors. And now let's talk about some immunological factors. Immunological factors causing DLE or not really SLE, right? Immunological factors. So you must have heard about the term tolerance. Tolerance is basically a method in the body which is trying to prevent disease by our own cells. Basically, tolerance is preventing. Tolerance, which can be provided by B cell or T cell, tolerance is a mechanism which is preventing autoimmune disease. But here in SLE, what must be causing it? What must be causing SLE? If there is a failure of tolerance, failure of B cell tolerance. Failure of B cell tolerance will cause autoimmune disease. Easy. Okay. T cells, just say T cells have a receptor on their top. T lymphocytes or T cells have a receptor on their top which is called TCR, T cell receptor. Very simple, not rocket science. This TCR is basically helping to identify the self pathogens. If there is a self antigen or the own cell of our body is basically attached over here, then this will have a mechanism that it will help in the tolerance mechanism. It will help to prevent the autoimmune disease. Okay. But here, if the TCR is rearranged, what I mean by rearrangement, molecules, it is a molecule, right? It will undergo rearrangements. It will not look like what it is earlier. Okay. You can say doglapan, right? That kind rearrangement. So it will not now recognize the self antigen. Okay. So you can write in the second part, so it will cause SLE, the rearrangement of TCR. You can write. So first one is the failure of B cell tolerance. Second one is the T lymphocyte rearrangement. Right? Okay. You know interferons? Interferons are secreted by viral infected cells. Okay. And we see that if there is a increase in type 1 interferons, it will also lead to SLE. Okay, done. So let me, so this slide contains all the factors which are leading to SLE. All the factors which are leading to SLE. If these all factors are present, then SLE will be caused. Understood? Okay. Let me quickly go through it. Then we'll move to the second part of this lecture, right? So what I told you, genetic factors, I told you about HLA-DQ, locus 150 and 0301. I told you complement factor deficiency. I told about the complement factor deficiency, deficiency of C2, C4, C1q. Immunological factors, I told you failure of B cell tolerance. Also, I told you rearrangement of TCR, right? And also increase in type 1 interferons, right? Then in environmental factors, I told you two things. One is the UV light exposure. Okay, UV light exposure, ultraviolet light, you just remember. And drugs, I was not talking about this type of drugs. SIP drugs, I was talking about SIP. So we have sulfonamides, hydralazine, isoniazid, and procainamide, right? This isoniazid is an anti-tubercular drug. Okay, isoniazid is an anti-tubercular drug, and this alpha drugs, hydralazine, these all are useful. Hydralazine, K plus open and all. Just don't need to go into that. Okay, so this was the etiology of SLE. So whenever a person asks you what is the factors causing, they even write here etiology. Etiology. So whenever they talk about your etiology of SLE, then you have to write these things. Understood? Next, move on to the these things are there. Okay, these are the features. Let me first tell you about the pathogenesis. Pathogenesis means how SLE is caused. We all saw this type of factors. We all saw these factors, but we have to understand how these factors are causing the disease, right? How these are causing the disease. So we have to understand the pathogenic model of SLE. Person. So this is Chintu, right? So Chintu was playing and got exposed to UV light. Was playing in the sunlight. UV light. UV light exposure was there. Whenever there is a UV light exposure, the body will try to kill the cells. The cells of the body will be killed, and killing of the body cells are called apoptosis. So there will be apoptosis. There will be apoptosis. And if there is apoptosis, then you all know that apoptotic bodies are formed. Basically, these are cell debris, right? The remnants, apoptotic bodies. I am writing AP. Apoptotic bodies are formed. This must be cleared out of body. These apoptotic bodies must be cleared out of body, right? We don't want this. But what it happens, this apoptotic body, who had the task to kill this apoptotic body? The complements. The complements. But what we have here, there is the complement factor deficiency, right? So we don't have complements. Okay, you don't have complements. Who will complement you? Still roasting. Okay. Apoptotic bodies are not cleared, so this accumulate. If they accumulate, the body will think of this as its own, or the body will think that it is foreign. Option 1 or 2. Yes, the body will think that we generally have the mechanism to clear the apoptotic body. Body is not thinking, it is direct complement is acting, is cleared. But here, what is happening? It is accumulating. The body thinks that it is its own antigen. Okay. Just think, the body thinks of it as a antigen. So it will like invite the T lymphocytes. Okay, the body will invite the T lymphocytes to kill the antigen. Apoptotic body binds over here. But what I told you, T lymphocytes have this TCR rearrangement. We discussed now. This TCR are rearranged. This mechanism is gone. T lymphocytes will not kill it. T lymphocytes will consider this antigen as foreign antigen. Foreign antigen. And this, if this is considered as foreign antigen, then there will be antibody production, of course. If an antigen is considered foreign, if a bacteria is coming to you, okay, it is foreign. The body will produce antibodies. The body is producing antibodies. Body is producing antibodies. We also have increased interferons, remember? These are also producing antibodies. And these antibodies will have the antigen bound, right? This apoptotic bodies will be bound to this. Okay. These are also, and interferons are also excess. More antibodies, more interferons, more antibodies. So this is a cycle. So this antigen antibody complex, mark my words, are basically traveling in the blood. This antigen antibody complexes are basically traveling in the blood. We call it circulating antigen antibody complexes. These are circulating in the blood. And if these are circulating in the blood, what type of hypersensitivity reaction you get reminded of? Yes, we call it type 3 hypersensitivity. Antigen antibody complexes circulating in the blood is basically called type 3 hypersensitivity. Done? Okay. Up to this is fine. Our body is handling. What happens? You have such. The girls wear hairband, you saw. So they also use hairpins. How do you know you are a boy? Okay. So these are hairpin shaped vessels. Hairpin shaped vessels. This antigen antibody complexes are basically, you must have read physics, right? If this was a surface, car was very speed at this point. Car got slowed at the topmost point. Then also it gets sped up as it is moving against the gravity. Then it is stopping it a bit, then going down. You must have read circular motion. Speed was lowest at this point. You must have heard. Okay. If you have had a circular motion, that is very important, very useful. So antigen antibody complexes got deposited here. Got deposited here. These things, these things, antigen bound with antibody, got deposited in hairpin shaped vessels, right? So this deposition is causing an inflammation, is causing the damage to this endothelium, is causing the damage to this endothelial part. Okay, and this damage is called as vasculitis. Vasculitis is a very important complication of SLE. People don't understand why there is vasculitis, right? Vasculitis. So this picture shows the vasculitis. You can see there is a damage to the endothelium. There is damage to the blood vessel. This type of spots are there in the body. Okay. Oh, so what this old domain is trying to say? What this picture is trying to say? Why there is deposition like this? Why this type of things are there? Let me tell you. You have skin. Of course, you have. This is the epidermal line. We have some part infoldings here. This is the epidermis. This is the dermis. Right? And here you have capillaries in dermis. What I will tell this dermal capillaries. Dermal capillaries. So what happens is that these are also hairpin shaped. Can you observe hairpin shaped? Where there will be deposition in the uppermost part. Here you have deposition. Here you have deposition. Okay. So deposition occurs in the tip of the dermal capillaries, and if you look from above, then what they will appear like? What a person will say, I got rashes, right? So here you have this type of rashes in the face. You have this type of rashes in the face. Basically, they involve the cheek, they involve the tip of the nose. This folds, this these are the lips. Lips means labia. We have the term labia, and nose means naso. So this portion, just pay attention at this portion, this whole part. These are called as nasolabial folds. Do SLE involve nasolabial folds? No. So the rashes, basically, just a minute, this is a call. Okay, so I'm back, and I was telling you that whenever, just a minute. So I was telling you that these rashes basically have a characteristic deposition. Characteristic, you can in your childhood times you must have drawn butterflies, right? Things like this. So let's assume this was the nose and these are the cheeks. Okay. So this appeared like a butterfly type of rash. So this rash appears like a butterfly. So these are also called as butterfly rash or SLE rash. And what is the characteristic thing about SLE rash? Characteristic thing is that these rashes spare nasolabial fold. So I told you what is nasolabial fold. This was the nasolabial fold. So these rashes are sparing the nasolabial fold, right? And this type of butterfly rash, SLE rash, or these are also called as, this name is very, very important. Just remember this name, malar discoid rashes. Malar discoid rashes. Okay. Can you remember? Easy? Yes. Malar discoid rashes. Okay. So these three are names. So I told you about vasculitis. I told you about what is the deposition of rashes. Next, we have something called as the diagnostic criteria for SLE. We have diagnostic criteria. Diagnostic criteria, what do you mean? Means how, as a doctor, you will analyze that a person has SLE. You can't say from these findings because these are non-specific. You can have many diseases with this type of findings, right? So you have to just say that we have a diagnostic criteria, and to remember this diagnostic criteria, just you have to look at these three things. Okay, what is this? It's the soup. What is this? A brain. And this is a doctor. I am telling MD. You know MD, after MBBS you do MD in many subjects. So brain MD. This is the diagnostic criteria. Okay. So you can write in diagnostic criteria you have soap brain MD. Soap brain MD. What do we mean? Okay. Soap. Let's start with SOAP, and brain I am writing like this. Brain MD I am writing just MD. Okay. So S. S means serositis. Serositis means serous. You know serous and mucus. Okay. So in serositis, you can have the inflammation of the pleura and the pericardium. Pericardium is covering the heart. Covering the heart is a pericardium. Covering the lungs is a pleural lining. So if these linings are inflamed, inflammation in this type of linings because serositis, you can have pleuritis, inflammation of the pleura, pericarditis, inflammation of the pericard, serositis. Done. Serositis is done. O. O means oral, nasal, and nasopharyngeal ulcer. Oral, nasal, and nasopharyngeal ulcers. Yes. A. A means in your joints. Basically, if this complexes are going to deposit in a joint, is going to deposit in some joints like a knee joint or your elbow joint, any joint, then this will cause pain. This will cause pain. Okay, this is called arthralgia. Arthralgia means joint pain. Arthra term is used for joint. Algia means pain. Okay, arthralgia. P. P means photosensitive rashes. Photosensitive rashes. Okay, clear? You know soap now? Yes? Okay. B. B is for blood abnormalities. Of course, If the immune complexes are moving in the blood, then you must have blood abnormalities, hematologic problems, right? What problems can you have? You can have anemia. What are the things you know in blood? Anemia, pancytopenia, you know. Okay, you know about thrombocytopenia. One knows, right? Hematologic problems, blood problems. I'm sure you must have understood. R. R means kidney problems, renal problems, renal problems. Okay. So in renal problems, you will find in the urine, you will find something like this. What are these things called? Can you tell me? These are made up of RBC. RBC casts. These are called RBC casts in urine, protein in urine, protein in urine. These findings are there. Protein in urine. Protein in urine. What are the? What is the you know you know constituent of protein? More than 0.5 gram per day. This much protein will be there. More than 0.5 gram per day. Okay. So protein in urine, proteinuria, we can tell. A means autoantibodies. Autoantibodies, of course, I will tell you what are the types of autoantibodies which are there. Just pay. Just wait a bit. Okay. Blood abnormalities, renal problems, autoantibodies. I means immunofluorescent findings. Immunofluorescent findings means antibody. This if you do immunofluorescence, you will get this type of pictures. This type of pictures are immunoelectron microscopic pictures. Okay, so these are immunofluorescence. Okay, means a dye is injected, and if the antibodies or some DNA is present, then this will have a illumination. This will have a illumination. These are immunofluorescence findings. Immunofluorescence, right? N is for neurological manifestations. Neurological manifestations. So you can see many manifestations are there. So it is a systemic disorder. It is a disease of skin. So not of the skin. It's not a disease not of a blood. Is a disease not of your CNS. It is a disease of total body. It is a systemic disease, right? Neurological manifestations. You can have seizures, seizures, right? Okay. And MD, can you guess? Yes. Malar discoid rashes or butterfly rash or SLE rash, right? Okay. So these are the uh, you know 11 criteria to diagnose SLE. Okay, 10 criteria, 11, yeah, 9 and 10. Malar discoid rashes I consider one. Okay, no problem. 10 criteria to diagnose SLE. But do you need all these criteria to say the person has SLE? All these criteria you know? No. You only require more than equal to 4. You only require more than equal to 4 criteria. If any 4 of these criteria, of these 10 criteria are present, you can successfully say that the person is a patient of SLE. So what is the cutoff limit? More than 4. 4 or more than, right? Okay. So this slide encompasses all the manifestations and the diagnostic criteria of SLE. Examiners have loved this question. Really. Examiners usually love this question to ask. The HODs and all, they usually ask you this diagnostic criteria. Okay. And it's a very, very famous question in the pathology as concept and medicine. So these things are there. Let's move on to another very important part, and this is the last part, that is called as immunofluorescence findings. Immunological findings or immunofluorescent findings. Immunological findings. Okay, immunological findings. Before starting, let me make you familiarize with the images. So these are the cells. This round one are the cells. Okay. So can you see the whole cell is shaded like this? The whole cell is uniformly shaded, uniformly shaded, uniformly shaded. What can you call the pattern? It is a you can say a homogeneous pattern. The first one is the homogeneous pattern. First one is the homogeneous pattern. Okay. You can see this looks somewhat like this, like some ghosts that all are ghosts. Okay, you get in WhatsApp like this pattern. What do you call this pattern? This pattern you can see here. This pattern is called the speckled pattern. Speckled. This is the two. Homogeneous pattern is done. Speckled pattern is done. Right? Okay. This dotted type of pattern. This dotted type of pattern is called as a centromeric pattern. We have five patterns. Out of the third one is the centromeric pattern, dot type of pattern, multiple dotted pattern. Right? This type of pattern in which only few dots are there. This type. Only few dots and generally dark type of pattern. This is called as the nucleolar pattern. Nucleolar pattern. Fourth type of pattern, nucleolar pattern. Right? And another type of pattern, this pattern in which you can see a rim only and the middle is pale. The dark, the luminous part is the ring only and the inside part is bit pale. This is called as the rim pattern. Rim pattern, and this is the tip pattern. Can you see the patterns? Homogeneous, speckled, centromeric, nucleolar, rim. Done. Okay. So what are these patterns basically? This homogeneous pattern is given is seen in antibodies against some things. Okay. This H. So this homogeneous pattern is seen when we have antibodies against this deposit in this type of pattern. Antibodies against H for histones, and chromatin. So whenever a cell has histones and chromatin, this type of pattern is seen in SLE. Okay, done. You can remember H and C for this high court. Histones and chromatin, H for homogeneous, H for histones. Okay, done. Speckled pattern, ghost type of pattern. Okay, ghost type of pattern. A ghost is a funny entity or a serious entity. It's a serious thing. If you see a ghost, it's a serious thing, right? You will get frightened, of course. The serious one. Serious. So I am highlighting S, R, and S. Okay. So whenever you have antibody against Smith antigen, Smith antigen, you just remember Smith antigen. Okay. You don't have to go into much detail. Or ribonucleoprotein or SSA and SSB. SSA and SSB, that is Sjogren syndrome antigen A and B. That is Sjogren syndrome antigen A and B. Wherever you have this type of things, will always get a speckled pattern. You will always get a speckled pattern, right? Easy and speckled. I told you looks like a ghost. Ghost is always serious. Easy to remember. Centromeric pattern. Okay, centromeric pattern. Centromeric pattern is seen in one condition, that is systemic sclerosis. Systemic sclerosis. Easy to remember one thing. Centromeric, centro means systemic. Okay, sounds like same. Okay. Nucleolar pattern. Nucleolar pattern. This is seen in, you can remember, mm, RN, RNA, I'm writing RNS. N is for nucleolar pattern. Okay. So whenever there is an antibody against RNA, antibody against RNA, and also in systemic sclerosis. So this is one condition, this is two conditions. In these two conditions, nucleolar patterns found. Nucleolar pattern is found. Okay. Fifth one is the rim pattern. The rim pattern. A rim pattern, uh, let's say this is the light area and this is the dark area. So whenever you draw a cell, what is the outermost layer? What is the outermost layer you draw? So let's say this is not a cell, this is a nucleus. So what is the surrounding nucleus? What's the layer surrounding nucleus? Nuclear envelope, right? So this pattern is basically seen when against antibody against nuclear envelope. Antibody against nuclear envelope. And also you can remember, and against antibody against dsDNA. Two parts. Antibody against nuclear envelope and antibody against dsDNA. You have rim pattern, and this rim pattern is sometimes some people call it peripheral pattern. Peripheral pattern. Rim pattern, peripheral. Done. So with that we come to the end of the SLE. This 45 minutes, I am sure you must have enjoyed it. I had taught SLE like you must never have read this type of content. Okay. This much is a minimalistic part. Okay, I have not gone into much detailing. I have not compromised on the content. It is apt for you for the second prof examinations and for NEET PG for handling questions of SLE. If you want to learn more in medicine part, then do comment. I will make another video with some extra things on SLE. This much is like apt. Okay, this is apt for you. This much knowledge is that I have not included the management, the treatment part, that things, because miscellaneous things. This is of course a rare condition, so not that common condition. Drug induced SLE and all these things are taken care of by doctors, right? So this much is done. This content is enough. And I'm sure you must have enjoyed in this session. If you have enjoyed, do like and share this session and the video, not the session, the video. And if you want to connect with me, then you can just follow me on Instagram, right? And ask your doubts if there are any. But I like usually have a lot of messages to reply, so I don't get time. But don't worry. And if you have to connect in Telegram, then you can search for Medbus by BT. Okay, this is a Telegram. Right? And if you have any doubts in the, of course, you can ask in the comment section. Okay. So that's all for today's session. Thank you and all the best. Bye bye.